So after this information, we come back to the content, to the heart of this discovery plenary session called Reality and Promises of Epigenetics for Animal Science, which is composed of 4 talks separated in the middle by the presentation which will be given by the recipient of this year's EAAP Leroy Award, Takehome. We gave this award yesterday night to Takehome. I remind you that this award is the most prestigious award given to scientists by our federation. Thus, this title of this discovery plenary session, Reality and Promises of Epigenetics for Animal Science, wishes to open a large window on what it is known on this area in animal models, in humans, as well as in farm animals, and what could be the consequences of such knowledge on animal selection and husbandry. For the first time also, this presentation is filmed and will be available in a few weeks in the EAAP website, but only for individual members. If you are not individual members, you can become easily and very likely at no cost. You can check how to do that in our website. So this is a discovery session. So please, in the room, dream a little bit when watching and hearing the speakers, and prepare your questions. We will have a section of questions after each talk. Thus, we'll have these 5 main talks this morning separated by coffee break. So the first speaker, that I call, is Brian Diaz, a renowned scientist from Psychiatry and Behavioral Science of the Emory School of Medicine in Atlanta, USA. He will speak about ancestral influences on descendant generations, and will take the example of the olfactory systems in rodents. Please, Brian, you have the floor.
Can anyone see that laser pointer? I don't think so, yeah. Okay. Good morning, everyone. I'd like to begin by thanking the organizers, and especially Andrea, Eleonora, Dorota, Megan, and everyone else for giving me the opportunity to come to Poland and present my work in front of you all. Since the focus of this session is, I'm going to skip all of that in the interest of time. Since the focus of this session is the reality and promises of epigenetics for animal sciences, I wanted to break that down, give you the take-home message from the talk, and then unpackage that. So when I talk about epigenetics, and I think it's instructive to have some broad framework in what we mean when we call epigenetics, I'm talking about something that is extra-genomic. So something that is beyond the ATGC of the genome. And when we talk about epigenetics, I mean the regulation of gene expression that comes about by adding acetyl groups, for example, to histones, which are bound to DNA, and that in some cases will increase the expression of genes. In other cases, the methylation of histones will decrease the expression of genes. And then in several cases, you can have methyl groups that are added to specific Cs and even other As in the genome that then allows for gene expression to be altered. So here in E, for example, you have methyl groups which are being laid down by something called DNA methyltransferase. For the most part, that methylation of the genome is going to shut down a particular gene. You're not going to have that gene expressed. But when you have the TET expression, which is in F right now, where you have the methyl groups being removed and another methyl group called hydroxymethyl being laid down, you have an increase in gene transcription, and so the gene is expressed. And that's going to be important as I go through my talk, which is why I bring it up here. So for the take-home message in terms of the reality and promises of epigenetics, here's what I think we know. The reality is that genes and environment interact. Epigenetics presents a mechanism by which the environment can lay down certain marks on the genome, and then those marks will allow for gene expression changes and then subsequent effects on behavior or any traits that you're interested in. What I'm going to show you is that stressful experiences interact with this particular genome. as a result of which you have profound influences on behavior and other traits because of these stressful experiences, because they affect gene expression via epigenetics. I'm going to throw in an unknown here, which is we know that stressful experiences affect the genome. We do not know how often and whether positive environments also interact with the genome, and that is thought of as future research. The promise of epigenetics, I think, should be very apparent to this audience in that we should be able to manipulate gene expression in the sperm and the egg to affect behaviors and traits. I say that this is a promise, but I also think it's a challenge. I come at this question from a neuropsychiatric point of view. So I'm in the Department of Psychiatry at Emory University, and I care a lot about neuropsychiatric disorders. So when I talk about gene by environment interactions, what am I talking about? I'm talking about the terrorist attacks of September 2001, and the work of Rachel Yehuda's group, who showed that babies who were in utero at the time that their mothers directly experienced those terrorist attacks, those babies have lower cortisol levels at age 1. And the same group has shown that descendants of Holocaust survivors had lower cortisol levels, which are predictive of PTSD-like symptoms later in life. I'm also talking about domestic abuse. And this is work of a colleague, Tanya Jovanovic at Emory, who's shown that children of mothers who were abused, and that abuse could have happened even before the child was conceived, those children are now more anxious compared to the children of mothers who were not abused. And then I'm also talking about instances of nutritional famine. So for example, the Dutch hunger winter of 1944 or the Swedish famines in 1968 have been shown to affect the metabolic status not only of the generation that experienced that trauma, but also several generations downstream, or future generations. And so the question then in the laboratory for us is, how do descendants inherit information from ancestors? And this is where I think it's important and becomes relevant to this audience. So in the animal sphere, I'm going to highlight studies from the nutritional spectrum and studies on the behavioral spectrum. So here I'm highlighting two studies. The first is by Margaret Morris's group in Australia, where they manipulated the diet of male rats and then looked at the F1 generation. So just as part of nomenclature, you know this, but the F0 generation would be the rats whose diet is manipulated. The F1 generation is the offspring or the future generation. And what they showed is that manipulating the diet of the F0 has profound consequences for the metabolic networks in the F1 generation. And then there's the work of Oliver Rando from Massachusetts, Worcester, who's shown that, again, manipulating the diet in the male mouse here. And this diet does not have to be manipulated when the female is pregnant. These are male mice. These diet is being manipulated even before the next generation is conceived. And what he's shown is that, again, there are profound differences in how the future generation processes their food and how the future generation metabolizes their food. From a neuropsychiatric perspective, I'm going to highlight two studies here. The first is the study by David Cruz, who took Michael Skinner's model. And what they did is they gave female rats a fungicide called vinclozolin. And they've now gone on to show that at least till the F6 generation, there are profound effects on offspring biology. That is, these offspring are more anxious, they have deficits in mate preference, they also have a slew of depression-like symptoms. And then the study on the bottom is a study by Eric Nestler's group, where they take a male mouse and they allow him to get beat up on by another male mouse. So that's called social defeat in a mouse. And what they find is that the future generation of this mouse that has been beaten up are more anxious and more depressed compared to the offspring of mice who were not subjected to social defeat. What was interesting is that some of their studies persisted even after in vitro fertilization. So that suggested that something in the sperm was changing to have an effect in the F1 generation. So with all of this work, what I want to emphasize is that where does one even begin to look for imprints of these ancestral perturbations? And to me, it's a little bit about trying to find a small needle in a giant haystack. So this is where I present the olfactory system as an alternative, another mechanism to study ancestral influences on descendant biology. And the reason for that is, and this work won the Nobel Prize in 2004 and has been shown to be conserved all the way from rodents even into humans. And so for your species that you're interested in, this is pretty much how they smell. So shown here in A is there's the skull of the animal, of the rodent, and there are olfactory sensory neurons which reside in the nose. So that would be in the epithelium right here for you and I. Those olfactory sensory neurons can express 1 of 1,000 different odorant receptors. Okay? That's important. There can be 1,000 odorant receptors that could be expressed, but every sensory neuron expresses 1. So, for example, in B, the receptor in red is expressing odorant receptor A only. And then what's interesting is that all olfactory sensory neurons that express a particular receptor, either red, blue, green, purple, they all send their axons into the brain, and those axons coalesce into a particular unit called the glomerulus, as shown at the bottom right of the screen. So each of those circles is a glomerulus in the olfactory bulb. Why is this important? This is important because in the laboratory we have a mouse called the M71 LAC-Z mouse. What we can do with this mouse is we can stain every neuron that expresses the M71 olfactory receptor 151 gene as blue. So now we can visualize from the nose extending all the way into the brain all neurons expressing M71, and more importantly, because M71 is activated by acetophenone, a specific odor, but not by propanol, we can ask, how does acetophenone exposure affect a particular population? So what we did was pretty simple in retrospect, but I think the findings are pretty remarkable. We conditioned an F0 generation to an odor, and by that I mean either acetophenone foot shock, acetophenone foot shock, acetophenone foot shock. The foot shocks are mild, the animals survive, but they do become fearful of acetophenone. Or we give them propanol foot shock, propanol foot shock, propanol foot shock. Then we wait 10 days to allow for the gametes to maybe get imprinted by this information, and then we allow for mating and test the descendant generations. What this allows us to do, remember now, is that if I use acetophenone, I can visualize acetophenone responsive M71 neurons. Therefore, our hypothesis was maybe the olfactory receptor gene that encodes for M71 might be bearing the imprints of this ancestral perturbation. So that haystack that I was talking about, the genomic haystack, to my mind, just got a lot smaller, and that needle that we were looking for just got a lot larger. So this is the experimental framework that we're using, and what do we find? Just a couple of slides with graphs. I promise not to bore you with them. I'm going to breeze through them. What I'm showing you here is behavior of the F1 generation that has never been exposed to acetophenone or propanol before. Shown on the y-axis is something called a startle to acetophenone. If you're in a dark room and someone does this, you're going to startle. So will a mouse. So will a rat. If a mouse or a rat is presented with that stimulus, along with a stimulus that has a relevance for it, it's going to startle a lot more. And so shown here on the y-axis on the left graph is percent startled to acetophenone. And what I hope you appreciate is the F1 generation in the middle, whose F0s were conditioned to acetophenone. They startle much more to acetophenone compared to the F1 generation whose F0s were conditioned to propanol. And there's a specificity of this response, as shown on the right-hand graph, where you have the F0 generation, when the F0 generation was conditioned to propanol, the F1 generation then startles to propanol, but not to acetophenone. And so there's a specificity that they have a behavioral sensitivity to acetophenone or propanol. Never you're not going to get me to say that these mice are fearful of acetophenone or propanol, no matter what you may have read in the press. They're more behaviorally sensitive to it. We are not saying that they are fearful of it. So then what we can do is we can also look at the geography of these M71 neurons in the brains of these animals. When an F0 animal sires offspring, and this F0 animal is not perturbed, you have the F1 generation glomeruli looking like this. When you have the F0 generation conditioned to propanol, and propanol does not activate M71, does not activate these blue neurons, you have no real change in the topography. But now, when the F0 generation is conditioned to acetophenone, and I look at the brains of the F1 generation that has never smelt acetophenone before, this is what the glomeruli look like. It's like the brain is devoting more real estate to processing acetophenone that had a salience for the ancestral generation. So we got these effects, and we were wondering, how is this happening? And there are two possibilities. There could be rearing effects in terms of mom might be provisioning maternal care differently when the father has been conditioned versus not, or there could be an inheritance of effect. So we did 3 experiments to tease this possibility out. We used in vitro fertilization, we looked at the F2 generation, and then we performed cross-fostering studies. I'm only going to show you the IVF data because I think it's important for the next part, for the main message of my talk, but suffice to say the data were congruent across all these particular possibilities. So here what we did is we fair conditioned the F0s and then just took sperm from these F0 males. So there was no mating, nothing of the sort, no traditional mating. And then we performed in vitro fertilization and artificial insemination, in vitro fertilization of females. And what we find is at the level of the neuroanatomy, once again, the F1 acetophenone animals, who are now generated from sperm from F0 acetophenone animals, have larger glomeruli, again devoting more real estate to that particular odor. And so this implied to us that there was something in sperm that was carrying that particular information. Okay, what in that sperm specifically? Remember, I started off this talk by talking about epigenetics, and I talked about histone acetylation and methylation, but I also talked about DNA methylation. We did not find any changes in histone acetylation or methylation in the sperm of these F0 animals. But when we sent the DNA from these animals to look at DNA methylation around these loci, what we found was pretty remarkable. Shown on the top is percent methylation in the sperm at this particular locus, the OLFR 151 or the M71 locus. And what I hope I've shown you is that there's a large amount of methylation when the sperm are got from F0 propanol animals, because these genes should not be expressed. But now in the F0 acetophenone sperm, you have less methylation at that particular locus compared to that control group, thereby potentially setting up the next generation to express more acetophenone. As a control, we looked at another olfactory receptor which is not activated by acetophenone or propanol, and there's no change in OLFR 6 between the groups. So what I've shown you then, encapsulated in this commentary which Moshe Ziff put out for us just before our paper came out, is you can take a particular odor and administer tiny foot shocks, mild foot shocks, and the animal startles to that odor and has a higher representation for that odor. That would be in the F0 generation. What we were able to show is that the sperm of that animal bears imprints of this ancestral olfactory experience, such that now the next generation has an enhanced representation for that odor and also startles more to that odor. And then the data I'm not showing you here is we found those same methylation changes in the F1 sperm. So why does all of this matter? So I hope what I've done thus far then is shown you that the environment, in this case olfactory experience, interacts with the genome, the OLFR151 gene, at the level of DNA methylation to potentially set up and influence behavior in the next generation. And I've shown you then that this was a stressful experience that interacts with the genome. The examples that I showed you, chronic social defeat in the mouse, the nutritional status, the nutritional challenges, the fungicides, all of them you would consider negative experiences. The unknown, as I said at this point, is do positive environments also interact with the genome? And that's something that we're really playing around with, but I'm sure you can contribute much more to this discussion than I can. But what do we do with this particular information? Well, there are two real questions to answer to begin with. One is, how does any environmental manipulation impact the sperm and the egg? In 1938, August Weismann put out a theory which postulated that the sperm and the egg are immune to all the detrimental influences, as well as positive influences, I would imagine, that the environment throws at it. But there are more and more data that are coming to the fore that suggest that the sperm and the egg are bearing imprints of ancestral environments. How is this so? And then there's a process called epigenetic reprogramming where any marks that the sperm and the egg might have are really erased or scrubbed clean soon after fertilization. And how do certain marks escape reprogramming? Once we address these two particular questions, we'll be able to manipulate how to affect descendant generations or buffer them from ancestral traumas. So the two studies that I'm pursuing right now in my lab is how is information from the nose getting to the gametes? And for that, we're looking at cancer biology, because cancer biology says that there are tumors which release these little vesicles called exosomes. Exosomes we know to be packed with things like non-coding RNAs, etc., which could then affect the sperm and the egg. And so we're profiling exosomes, we're profiling the microRNA, the piRNAs, the tRNA fragments in there, and looking at what effect this might have on the sperm and the egg. And then we're also looking at within the sperm, on a genome-wide level, what are the methylation changes that are occurring. When we're able to do this, the question then becomes, can we manipulate gene expression in the sperm and the egg? Again, as I said, this is a promise. It's happening. I can take sperm from my F0 acetophenone animals, get only the non-coding RNAs, and inject those non-coding RNAs into sperm of naive animals and recapitulate my effects, right? I can use CRISPR technology. I haven't yet, but people have used CRISPR technology where you can go in and edit the genome at specific loci for any animal that you know the genome for. And people have done this in the human embryo recently. So it is the promise that comes with epigenetics, but as I said, it is also fraught with peril. Where does one draw the line in terms of what does one manipulate? But more importantly, we have no idea how that's going to affect the organism as a whole. All of these perturbations, non-coding RNAs, CRISPR technology, have what we call off-target effects. You might be targeting gene A, but you might also be hitting gene X, Y, and Z. And just because you target gene A, and in the event that you're supremely lucky and don't have any off-target effects, most traits are polygenic. You're obviously going to affect different genes downstream, and we don't have the bandwidth and the intellectual smarts right now to be able to deal with those repercussions. So this is the promise for sure, but it comes with a huge responsibility and a lot of peril. So with that, I'll just leave you with this reality, the unknown, and the promise that epigenetics has for animal science as well as neuropsychiatric disorders from the perspective of my work. And I'd be happy to take questions if we have any time. Thank you. Sorry, we're down time.
Thank you very much, Brian, for this excellent,
Sure.
exciting presentation and to be on time. So we have time for 5 minutes for questions. So please, if you have questions, either you can come to the microphones which are there, either you can take this microphone if somebody can pass it. I don't know if...
There's a question right there.
So first question, yes? Could you give your name and country, please?
It's on.
It's on, David. You can. It works. Yes, it works.
David Pethick from Murdoch University, Australia. Thank you for your talk. This is not a challenge to the statistics. It's more a question in your mouse model. I think you had maybe 12, 14 mice in one of the earlier slides. I was just interested to know, how consistent is the response? Do some mice not respond at all, and do others respond?
Ladies and gentlemen, I give you reviewer number 2 now. That's a really good question. So here's how I think about it. If you see the spread on our data, it's obvious that not all the mice are responding, and you would expect that to be the case. I think we're playing a numbers game with the sperm, and so not all sperm may be equally affected. I should also say that I'm not excluding any F0s from this analysis. And so you could have an F0 animal who did not learn about the conditioned odor for some reason. The shocker wasn't working or something of the sort. I've not excluded that animal and his offspring from future analysis. But I think your point is well taken in that in our future studies, we're going to do a correlation between the F0 behavior and then the F1 behavior as well as the methylation, and hopefully be able to pass out what that relationship is. And I think that's really important because that's going to affect how we look at these data.
Thank you. Second question, yes?
Bonnie Millard, University of Guelph, Canada. It's a somewhat related question. What were the age of your mice when you're conditioning them? And does it matter? Do young mice condition their epigenetic effects more readily than if you try and condition adults? So children versus adult, for example.
So, it costs me 60 cents a day to maintain a cage of mice, which might not seem a lot, but when I have 200 cages and budgets are tight, it's tough. So those are experiments that we'd like to do. For the paper and every subsequent experiment that I've done is 2-month-old adult males, because male mice will reach sexual maturity at 8 weeks. And then they're mated 10 days later, so I would imagine about 3 months is when we see these effects. With respect to do adults learn differently than, or juveniles learn differently than adults, absolutely. And the question or the little wrinkle with the juveniles is that juveniles actually develop what is called infantile amnesia, and so they will forget conditioning. And so that would present another challenge to whether this is actually a memory that is being inherited or merely a sensitivity. And I actually think it's a sensitivity. But we haven't played around with those conditions. In the grander scheme of things, in utero, I can tell you that if I do this in utero to mom, the fetus does not care about this odor any differently than a control fetus.
Last question, please.
Yeah, Marlene Pibamase, University of Guelph, Canada. My question is to do with your CPG methylation. In terms of the sites that you looked at with your target gene, did you see a variation in the CPG methylation between the different CPGs, or was it consistent across that area?
It was remarkably consistent. So if I'm not mistaken, there was a CPG14 at one particular part of the gene, and across animals, we saw that CPG really affected. Now, I think the cancer folks and other people who study DNA methylation will ask, what does a single change in a CPG site actually mean for gene expression? There's some people who've shown that a single CPG site can dramatically affect transcription of a gene, depending on where it is in the gene. Which is why we're taking a step back and actually getting a bird's eye view now with genome-wide methylation and actually looking for clusters in promoters, CPG islands, CPG shores. And then if there's a consistency there, I think we're in a better position to answer what effect that will have on gene transcription.
So we'll end with that question. Thank you very much, Brian.
Thank you.